Good morning everyone. I am Ragavi, working as an assistant professor in the Department of Biochemistry, JJ College of Arts and Science. Now we see about a topic in the immunology, that is a technical topic, enzyme linked immunosorbent assay. It is shortly known as ELISA. Before we introduce the ELISA, we know the terminologist or the in the immunological topic, that is the antigen. It is shortly named as Ag, a substance that when introduced into the body stimulates the production of an antibody. It is known as antigen. That's why in an antibody, they are the large Y-shaped glycoproteins. They are produced by the human system to identify and neutralize the foreign object, that is the antigen. It is known as an antibody, that is shortly denoted as AB. Immunoassay. Immunoassay is a test that uses the antigen antibody complex. It is shortly known as the immunocomplex as a means of generating the measurable result by using the antigen and antibody. In an antigen antibody reaction, there are two types, that is primary antibody reaction and secondary antibody reaction. In a primary antibody reaction, antibody specifies to a specific antigen only. In a secondary antibody reaction, antibody specifies to a primary antibody itself. That is, the enzyme linked will convert the colorless substance to a colored product, indicates that the presence of antibody antigen binding complex, in which secondary antibody involves the enzyme. Now we are actually involved in the ELISA. ELISA is a method used in the immunology and other scientific field designed for the detecting and qualitating substance such as the proteins. In proteins, it may be a peptide or an hormone. In an antigen in general. The next one is the antibody. The principle of ELISA is to detect the specific antibody antigen reaction by using an enzyme which can convert a colorless substance to a colored product, indicating the presence of antigen antibody complex or the antigen antibody binding. Purpose of ELISA to determine the presence and concentration of a particular antigen or an antibody in a given sample. Thus, it can be run in a qualitative and a quantitative format itself. What is meant by a qualitative ELISA? It is a result of provides a positive or a negative result from the sample. It gives it is a positive result or only a negative. In a quantitative ELISA, the optical density or the fluorescent unit of the sample is implanted into a second curve that observed from the serial dilution of a standard. In qualitative, it gives the amount of antigen which have infected our body. In a qualitative, it justly involves the presence of antigen in our body. Application of ELISA. ELISA can be used in the field of medicine, food industries and in a toxicology labs to evaluate the presence of specific antigen or a specific antibody in a sample. They can be used for the screening the donated blood for the evidence of viral contamination or measuring the hormonal levels. It can be measures the autoantibodies in an autoimmune disease such as the rheumatoid arthritis. This is the plate where the ELISA has to be done, and the form it is known as the microtitrate plate where the sample are involved, that is the antigen antibody reaction actually takes place. Next we discuss about the type of ELISA. There are 4 different types of ELISA: direct ELISA, indirect, sandwich and competitive ELISA. First we move on to the direct ELISA. It is used to detect the presence and the concentration of specific antigen in the given sample. The test which is considered to be a simplest type of ELISA. Principle: the antigen of an interest is observed or fixed to a microtitrate plate. An enzyme is linked to an antibody applied to the antigen. The enzyme antibody will bind to the antigen of interest. By adding the enzyme substrate, the enzyme will convert the colorless substrate to a colored product. the colored product is proportional the amount of antigen of interest. The color itself indicates the presence of antigen in a sample. The name is known as a direct ELISA due to the antibody linked to the enzyme is directly binds to the protein of inserted antigen. In direct, we should note about the hiding and blocking the buffers which contains a non-reactive proteins to block all the onboarded site in the wells to prevent the fault or false positive result or non-specific binding. It is important to block the buffers in a given sample in a direct ELISA. In an indirect ELISA is used to detect the presence and concentration of specific antibodies. In an indirect ELISA, the principle is mainly depend upon the one more labeled secondary antibodies is added in the reaction. The primary antibody added to the fixed antigen, then labeled secondary antibody added to the recognize the primary antibodies. The color or the signal produced as a result of addition of substrate is proportional to the antibodies in the sample. We should note that in an indirect ELISA, the primary antibody is not linked to the enzyme and it is the antibodies of interest in this case is specific to the antigen. That is, in primary antibodies is named as first antibody, the secondary antibody is named as second antibodies is conjugated